Okay. Well, let's go ahead and finish chapter 10, shall we? And yes, we'll be finishing up chapter 11. So the exam, which is going to be next week, Thursday, will be probably 5, 10, and 11. Those three. So 11 being, we only have like a couple of slides left on 10, so most of today is going to be on 11. So we'll be able to get probably more than the day. And that's about where we've been landing in most of the sections. So let's just kind of sort of clean up a little bit of chapter 10, right? We'd already been talking about the two different types of genes, right? Tumor suppressor genes and proto-oncogenes. And we were going to talk about a couple of different types of tumor suppressor genes. The big one, the gram poobah, so to speak, of the tumor suppressor world is a protein known as P53, which has kind of a bit of an inglorious name, right? Basically, all that means is protein 53. That's all it means. So it doesn't have like some little fancy dancy schmancy name. Basically, when they were doing screens for different proteins that were acting like, that had the ability to suppress tumor development, they just basically picked up a bunch of proteins and they figured out this one was 53 kilodaltons. So this was protein 53, that was 53 kilodaltons. That's where P53 comes for. It turns out to be a checkpoint regulator, so a G1 checkpoint regulator. And so it's important. So this is kind of like a big monitor, basically, to make sure that everything is going right. Remember our two mantras in cell division, right? The first one is do it right or die. And the other one is do what you're told or die, right? Either one, it's kind of like, you know, a binary situation. So you don't really have much wiggle room there. So who's to say whether you're doing it right or not? Well, that's P53's job, right? So if you basically have a train wreck of DNA, like a bunch of mistakes and things of that nature, then basically what P53 is going to do is it's going to basically halt cell division. So it puts everything on hold, just like what you would expect from a checkpoint. And then what it does is it basically allows time for you to be able to repair your DNA, right? And that P53 has three primary functions to it, one of which is it's a stimulator and activator of DNA repair. So if you've got a bunch of damaged DNA, then this is the one that starts to turn on all those repair mechanisms to fix your DNA. The other thing that P53 actually does as well is it's a transcription factor for other cell division genes. And remember, we talked about transcription factor is just one of those regulatory proteins that either activates transcription of a gene or deactivates, right? A tumor suppressor was a perfect example of that. Your ability to either inhibit the gene expression or to stimulate the gene expression. That's what transcription factors have a tendency to do. And so P53 is like a transcription factor that affects something on the order of like 500-ish or so genes. So there's a lot of genes that P53 will drive. So it's basically in everybody's business, essentially. And then the third major thing that it does is apoptosis. What's apoptosis? That's cell death. So the trajectory is very clear. If you're a cell and you've got a bunch of damage in your DNA, then what's going to happen is P53 is going to pick that up. It's going to start, it's going to halt the cell cycle, and it's going to give you some time to repair that. So it's going to start turning all of those repair mechanisms and give you a chance to fix all of your DNA. Now, you usually have one of two outcomes occurring, right? So if you have basically a lot of DNA damage, then that suggests a couple of things. First of all, either you just got walloped by a lot of mutagenic stuff, chemicals, radiation, whatever, right? Like you just were on the business end of a nuclear bomb or whatever. That's one thing. The other thing is that life isn't going so well with you right now, right? So both of those typically, both of those scenarios, that is to say, you're trying to fix your DNA, trying to fix it, 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 but it's not getting fixed. What that tells P53 is like, oh, I see. So the damage that you have is not fixable. Is that what you're trying to tell me? Right? So you don't want to tell P53 that, because what is it going to do? It's going to default then to cell death, right? And so basically it's going to say, listen, you're taking way too much time on the clock. I gave you 5 minutes, right? You get yourself together, you get your DNA fixed, you got 5 minutes to do that. If it takes longer than 5 minutes, then one of those other two scenarios are probably what's at play here, right? Either you've got so much damage that we have to basically kill you because you cannot go forward as a genetic trainer that you are, or the DNA damage that you have is unfixable. Either of those is intolerable. Both of those, they'll then basically switch P53 from the fix it mode to the listen, this is a wash. So I'm ordering you now, stop fixing your DNA and take the knife to your throat, right? So that's the brutality of do it right or die. So that's basically what P53 does. And so obviously then, if you're a cancer cell, oftentimes, basically you are incurring lots of damage. And the last thing that you want as a cancer cell is to be told what to do. So what do you have to do? Well, just like the toddlers, right? The angry little toddler. What do? You take out the adult in the room. In this case, who's the adult in the room? P53. So as a result, oftentimes we see in cancers a mutation in P53. So the cancer cells have done just that. They've localized P53, they've identified P53 as the voice of authority, and they've taken them out. We see this actually in something like 50% of cancers that we recover. That basically means that taking out the adult in the room is a common strategy for cancer cells. Because once you take the adult out of the room, there's nobody telling you not to do something. You can do whatever you want, right? It's Lord of the Flies time now, right? You've got the island to yourself. You want to be a cannibal? Feel free to, right? Who cares about the conch? Just, I'm going to eat you, right? The conch now doesn't mean you get a right to speak. The conch now means that you are the next dinner, right? Okay, I got two questions, Christopher and Melissa.
So when you are cancer researchers are the
It's not that easy. In this case, there's actually a lot more adults in the room. So there's not just an adult with a room full of toddlers. There's like an entire network of supervision. And different members of that supervisory network can be taken down. It just happens that P53 is kind of a little at the top of the food chain. But there's a lot more going on there. Just like it was with the Lord of the Flies, right? They didn't just basically devolve into cannibalism right off, right? I mean, they kind of organized themselves in a little society. You had one group that wanted to maintain the order that they learned from their little academy school, right? Choose your leader, let's all be democratic, let's be civilized. And then you had another group that just wanted to spin off into craziness. And so it kind of took a little bit of time, right? So you kind of had different levels there. So you had these kids that still wanted to maintain order, just like here, right? Just because you remove the adult from the room doesn't necessarily mean there aren't going to be sympathizers to civilization and order, right? So there's a lot more going on there. Melissa, you got a question? Well, the cancer cells don't really know anything. When they're taking up P53, it's largely random. So one of the things that happens with cancers is that they tend to accumulate lots of damage and lots of mutations, but the mutations they accumulate are random. So it's kind of like they're trying to crack the safe to give them that magic sequence of mutations that's going to make them super cells so that nobody can touch them. They're immortal. They can live forever. They can divide as much as they want. They can kind of take over the world. That's what they're trying to do. But the problem is they need a sequence of favorable mutations in order to get to that. And so what they do is they're kind of like a crook trying to crack the numerical combination on the safe. I think, what was it? Was it Die Hard that did this, where they kind of had the guy with this little gizmo, he kind of stuck it up to the safe, and it was just like randomly going through like number options. It was like a little computer, just randomly trial and error testing all these different numbers, and eventually it'd lock one in. It's like, got one, right? But it was like a sequence of like 6 numbers, so it had 5 more to go. And so as soon as it got that one, it got a little stronger, right? And then you get the second one and you're a little stronger yet. And you get the third one and you're a little stronger yet. And then that's kind of what cancers will basically do. So they're just kind of constantly going through there. Now, as it turns out, one of those random favorable mutations that you get is P53. That's the reason why it's not in all of them. It's only in about half of them. But which actually, if you think about it, is basically a coin toss, which is randomness, right? Half of them flip heads, the other half flip tails. Okay, so let's go ahead and take a look at this. So this is kind of showing you how the growth factors actually feed into all of this sort of tumor suppressor and oncogene stuff. So here you can kind of see the growth factor. This is an example of PDGF, right, which is a receptor tyrosine kinase. So here's your receptor side of it on the extracellular side. On the intracellular side, you can see this is your kinase activity of your receptor. And so it has the ability to phosphorylate the beginning of what is this sort of nested hierarchy of rolling kinases. So you're just kind of turning on a protein that turns 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 on a protein that eventually turns on something you want, right? So it's like this multiple layers of regulation. It's like great government bureaucracy, right? So in this particular case, the growth factor will turn on a protein called RAS, which will activate RAF, which will then phosphorylate and activate MEK, which will activate ERK. And ERK, you don't have to memorize what these names mean, right? But ERK just basically goes through and into the nucleus, whereas a kinase, it will actually phosphorylate a protein that is kind of a dimer at the moment of RB, retinoblastoma, and E2F. So what it does is it phosphorylates retinoblastoma, which basically breaks the two apart. So retinoblastoma will go off and be a transcription factor for lots of different genes. And E2F itself will also be a transcription factor for a lot of cell division genes, like cyclins and other proteins necessary in S phase. Normally it is off, but you turned on the function by phosphorylating it. Now, I don't want you to memorize this whole thing. What I want you to do is pull out the big broad brushstrokes, right? Notice, first of all, what you have is multiple layers. From the initial signal out here to the initial activity down here, you've got several layers in between. It's like a grapevine, right? Tell your people to tell their people to tell their people to tell their people to tell their people to tell a final person to do their job. Right? That's kind of what it looks like. Well, any one of these, like for instance, if they get mutated in sort of a stuck on state, like perpetually active, can act as an oncogene, because they'd be sending perpetual signal to turn on whoever my target is, right? So any one of these guys could act as proto-oncogenes. And a proto-oncogene, basically all it is just a cell division gene that if it mutates can be a problem. That's all that is. It's a normal thing. A proto-oncogene is not abnormal, right? It's the oncogene that's abnormal. Oncogene, as in oncology. So the other thing also to notice is that these guys down here represent the tumor suppressors. So one is in the transcriptional activators. So basically, these guys will then do their job. So by the time they get the message, it's gone through many, many hands. Okay. And so a couple of things also to kind of point out here, because this is the way signaling works in the cell. Whenever one cell communicates with another, this is what happens. So you've got this really big, complicated, multi-layered, multifaceted signal transduction system before you finally get down to the bottom of it, which is usually just like, would you just turn your transcription on? Right, which is a simple instruction. But a lot of times students will take a look at this and be like, well, wait a minute, why, if it's just such a simple instruction, then why do you have so many layers? There's a very good reason. The reason why you have so many layers is because before you turn retinoblastoma loose and E2F loose, because remember, these guys are turning on cell division genes, you always double check to make sure you heard that right. It's like, wait. Okay, so you're phosphorylating me, so you want me to turn all of my cell division genes on? Are you sure? Yes, I'm sure. Are you sure you're sure? Right? So you kind of keep asking them, and that's essentially what's happening with each one of these layers. It's like this gets the message to tell your people to transcribe. And it's like, are you sure? So let's go ahead and go to RAF I'm not going to just tell them straight off to do it. I'm going to send this on to Raf. Raf, you double check yourself. Are you sure? Right. So every single time you go through one of these layers, it's like a double check of the message. Are you sure about this? Why? Because what happens if you get the message wrong? Oh, yeah, no, I wasn't so sure about that. So now in retrospect, I wish I hadn't told you to do that. Guess what? That's catastrophic fail, isn't it? So this is like a life and death situation for the cell, which is the reason why you have multiple layers in there to double check it, right? It's just like the nuclear bomb, right? I mean, there isn't a button in the Oval Office. There never has been, right? Because nobody in their right mind would give one president, especially the last few that we've had, the full authority to just push a button and then off goes the silos, right? What happens is basically you have a system of checkdowns, right? The so-called football, right? The president says, I want to do this. And who has to confirm? Has to confirm that? The Department of Defense, right? The Secretary of Defense has to confirm that. So the president does it just like to say, let's, I'm in a bad mood. Let's just go ahead and blow up Paris, right? And I don't know. Yeah, it's just like, I don't want to pick on the French, right? I don't know why not, right? I mean, they're kind of central in Europe, so just it's easy to get to. So why not, right? I mean, if you're in a bad mood, doesn't really matter who you blow up. Just blow somebody up, you'll feel better until you realize you just destroyed somebody. And then if you have a conscience, you'll feel bad. But if not, then you're a sociopath. But which might explain actually several of our previous presidents. But anyway, so the idea here is you have a series of checkdowns. By the time a missile leaves the ground, multiple people have signed on to that decision. Why? Because it's too consequential to leave it in the hands of one person. Okay.
In terms of energy, the cell trying to do it as possible, why wouldn't it just connect? Does it technically the hub, or is it?
You mean like between the receptor and the final transcription factor in the nucleus? So spatially you're talking about.
Yeah.
So there's really not a spatial organization. They're just kind of all over the place. So they're just writing spatially just so you can see it clearly. I mean, in some cases there is some spatial tethering, but not all parts of the system are spatially located. Like for instance, there's some communications that are happening at the membrane and then some that get transferred inner to the, because ultimately what's going to happen is you have to go into the nucleus. Before you do that, it would be kind of nice if you actually manage to get yourself a little closer to the nucleus, so you're not having to sort of like, travel cross country to get there. So parts of that transduction system does have a spatial component to it as it starts to relocate its ultimate signal, get closer to its final termination point. But that doesn't affect how they turn themselves on and off and things like that. Melissa. No, because I mean, what you have is basically genes that I just blanket refer to cell division genes. So they basically make a protein that is involved in some way or form in the cell division process. And generally speaking, we identify oncogenes or proto-oncogenes as genes that are normally, right? And they're controlled and they're off. They're just like the Lord of the Flies. The kids, when they are in their private school, are under supervision and they're all orderly and disciplined and things like that. But if you allow them to go without supervision, they will go rogue, right? And they will turn into something very dark and malevolent, right? Like the cannibal kid in Lord of the Flies. Was Jake or Jack? Jack. He was Jack. Yeah. Great book. Read it. Don't watch the movie. Don't be lazy. Read it. Right. You learn how to read in the early grades for a reason. Use it. It's not so you can read Twitter. Right. So you can actually read real stuff. But anyway, they're like, huh? Books? What are books? Sorry, I'm just picking on the millennials. Which is like most of you in the room. But anyway, so I am outnumbered. Old grandpa fogey that I am apparently, but right. So proto-oncogenes basically are normal genes, but then when they get mutated, they have the ability to sort of transform into sort of this dominant, negative, malevolent type of a gene, and at that point it becomes an oncogene. And so that's when they get stuck in that on state, right? So it's a kind of that criminology theory that a lot of times there's more criminals than we think. It's just that most of them don't have the opportunity, right? So that's like a proto-oncogene, right? Like a criminal in waiting, if you will, is like a proto-oncogene. It's like, for the most part, as long as the construct of society and order maintains its integrity, they're never going to commit a crime. But many of us, probably a disturbing number of us, likely, if we thought we could get away with it, would probably commit some pretty heinous crimes. You know, it's true. We're not as noble as we like to think we are. We're just like maybe that far away from just being brute animals. Actually, animals are a little bit more noble than that. How do they become oncogenes? No, usually once you become something like a dominant negative, you're screwed. That's pretty much, that's your life. That you never get that one back. You can never go back from that one. You would literally have to do something really drastic, like for instance, CRISPR, where you have to literally go in there and like just completely replace that gene. Because it's not easy. It's not that you're missing something, and that's the reason why that's like a tumor suppressor. Tumor suppressor is running off the rails because you're missing something that's supposed to be there. So the idea is if you can go back and supply that thing that's missing, then we can get it back on the rails. But with the oncogene, it's not missing. Nothing's missing. It's just that you've just become a horrible gene, right? So that's a little trickier to deal with. That makes sense. So these guys are always stuck on, and of course, tumor suppressors are the ones that are recessive in nature. We've already talked about P53. The first one that we found was called retinoblastoma, RB, actually. That's a tumor suppressor we found first. And the way we found it is the way we find a lot of things. Is we basically found a bunch of little kids going blind, which is really tragic. What we noticed is actually retinoblastoma taught us a lot about tumor suppressor cancer development and genetics of cancer and things like that. But remember when we said that tumor suppressors are recessive, right? Which means that you need 2 mutations before it becomes a problem. Normally, most of us have 2 healthy copies of the tumor suppressor gene, one from mom, one from dad. Now, generally speaking, throughout most of our life, we're basically exposed to natural background radiation, and we're chemicals that are naturally mutagenic and stuff like that. There's just always a background level of mutation. And we're always accumulating mutations throughout our life. The problem is the accumulation of those mutations is random. So there's not a gene that gets preference or preferential treatment. It's just randomly, it's like you're just throwing mutational darts at your genetic dartboard, right? And so if you're really unlucky, it's going to hit your bullseye. And if you're really lucky, then it's just going to graze you a little bit, right? But the idea is because it's random and it's shot all over your entire genome, for us to pick up that first mutation takes about 2 to 3 decades. Sometimes 4 if we're lucky, right? It's all random. It just depends. If we're really unlucky, it could be less time. If we're really lucky, it could be more time. But guess what? It doesn't matter, does it? Because we have a healthy copy still. So as long as we have that healthy copy, we're good. So it takes another maybe 2 to 3 decades to pick up that second mutation, right? So at this particular point, if we take a look at the high side, how old are we? In our 60s, maybe. If we're lucky, maybe our 70s. And not only that, but even though we just lost our tumor suppressor now, it doesn't necessarily mean we just like, okay, let's turn the light switch on, you got cancer, boom, right? Cancer development takes time. All that means then is you don't have a cell necessarily that's gone rogue yet. All you have now is you've relieved the control from that particular gene. That gene going rogue may not be enough for the entire cell to go off the rails. So you may need a couple of others to go along with it And then slowly, even if the cell did go rogue, it takes the cell some time to slowly kind of break up, right? Because it starts off somewhat normalish, just like with Lord of the Flies, right? The kids didn't just go straight to cannibalism. They kind of started off, they were mostly normal. They're like, okay, so we're well trained and we're supposed to be good. So let's do what we're supposed to do and organize ourselves, right? But as you kind of started to get a little further away from that regulation and realizing it's like, wait a minute, so I'm not regulated, so I can pretty much do whatever I want. Then you kind of get a little bit more malevolent, and then you start to develop tumors. That could take years in its development. So even the moment you lose your tumor suppressor, it can still take quite a bit of time before it becomes a clinically diagnosable problem. So you may well die of old age before you get there. Two things. It's not as fast a process as we think, right? And number two is, this is also the reason why we see cancer more frequent in older people, why we associate it with an old person's disease. Because most cancers are random mutations that we accumulate in our body cells. And it just takes time to collect that many mutations. Yeah.
Just with all of the advancements in artificial intelligence, there have been a lot of like growth in the direction of using it towards cancer treatment, and like spotting early on. I know that it actually detected in a woman like well before it would isolate where in her body that it was the breast cancer was, and they were able to so like is that, you know, is there still a chance of continue, like obviously there's you still have cancer cells in your body somewhere else, but taking that out, like, does this kind of change the way that our body will continue with AI? Like, you know, how does that affect?
Well, first of all, two things. AI is scary as hell. For reasons that don't have anything to do with medicine. And the other one is, as long as you have one cancer cell, you've got cancer. And that's part of the problem with cancer. And that's always part of the problem. It's like, oh, well, we surgically removed that. So how come it's back? Because you didn't remove it. You remove most of it. But remember, cancer is like a zero fail mission. You have to get it all. As long as there's one left behind, in time, it will rebuild itself and it will come back. And that's the problem with that. AI is a fascinating, beneficial sort of thing. So basically, artificial intelligence is adaptive. It's adaptive technology. It's basically adaptive programming. So it's essentially, most of it's mirrored after our own, the way our brain works, right? It's basically technology that has the capacity to learn, right? Learn and grow. And the idea of AI with cancer is really cool because you have a cancer population that's constantly changing and morphing, right? And looking for an advantage. But then if you counter that with AI, which is constantly adapting and sort of doing this little chess game with the cancer cells, then the AI eventually can figure out the chess strategy that the cancer is playing and then outplay it. So that is an interesting angle. I honestly wouldn't sign up for that one. I'm sorry, I've seen too many movies perhaps, and I've seen too many videos. I don't know if you guys have seen the videos of some of these AI robots out there saying they're going to destroy humanity. I'm like, okay, and the scary thing about AI is not like, we can't fall back on like, oh, well, they only say what you put in there. Not with AI. AI synthesizes their intelligence, which is what's scary, right? So it's kind of bizarre, but I mean, when I was a kid and watching Terminator, I'm like, whatever, it's kind of cool science fiction. But then when I look at AI and I look at what they're able to do and the way their neural networks work and the way their adaptive software work, especially when you get two AI intelligences working with each other and the way they learn and grow and basically kind of out kind of think you, basically, is what they do. I mean, I have flashbacks of Terminator. It's like, oh, okay. Like, not the first one, but was it the one where they actually try to go back and eliminate the original creator of the AI, the black guy. Remember that, right? And they said like they had a day when it became conscious, and then that's when it started taking over the world. Yeah, it's one of the later ones. It's like 3 or something like that. I think that might be 3. That's the one that I liked, because it's like you're looking at that and you're like, okay, yeah, that's kind of, then it was science fiction, but it's funny because when you watch, like, especially when you watch all the engineers talking about it, and it's funny because the engineers are arguing with each other. So some people building the AI systems and they're turning them off now because they're like, okay, so you just totally came up with this on your own, that you think it's more efficient to just annihilate humanity. It's like, okay, that's not okay. That's not what you want your artificial intelligence to grow toward. Yeah. Which, if you think about it, go back and watch the Terminator series. That's exactly the way the Terminator saw things. It's eerie, actually. It's a little freaky when you kind of look at it. But then you see not just like, I mean, okay, so it's creepy enough to have a robot say they want to annihilate humanity. I mean, that's creepy enough. But then when you actually follow that up and dig into it, and you see like what they're able to do, how the intelligences are able to coordinate and communicate and grow and build. I mean, you can see it's like we've kind of got this nuclear reaction in our hand, and it's getting out of control very quickly. And we're not going to be able to like control it. It's going to be a little weird eventually. Anyway, okay. So we'll have nightmares about that. Maybe. Be like the Matrix, right? We'll all just become batteries. Yeah, all become batteries for the Matrix. Love that movie, by the way. Loved all 3 of them. Okay, let's move on to chapter 11. So in chapter 11, so we just basically took a look at cell cycle as a thing, right? Now in chapter 11 with meiosis, I don't really look at meiosis as sort of like, oh, well, here we're just going to be taking a look at another cell division strategy. What we're going to be doing here is I take a look at chapter 11 as the preliminary foundational work that we need for chapter 12, right, which is genetics. So that's basically our genetics chapter. And so in order to understand genetics and how we pass traits on, we have to understand a thing or two about the mechanism by which we do that and the purpose for which we do it, right? So this is our sexual reproduction and meiosis chapter. And this is it. We're finally here at the reproduction chapter, right? The semester's almost over. We're finally here. Yay. Okay. But let's take a look at this, right? Because not all reproduction is sexual reproduction, right? So you have sexual reproduction and you also have asexual reproduction. So what I want to do is have you guys spitball for me a little bit. Some pros and cons for each system. Now, with asexual reproduction, just to kind of give you the basics of that one, this is essentially like making a carbon copy of yourself. That's pretty much it. Versus obviously sexual reproduction, I'm assuming that, you know, we kind of have the basic nuances of sexual reproduction down for the most part, right? Several of us, most of us have probably tested out various parts of the theory. And so we're well acquainted, right, with what it entails. And so we're well positioned to talk about pros and cons of the system. Okay, so let's take a look at it. You can start anywhere you want. Give me some pros or cons, either asexual or sexual. Go for it. Yours. What? What? Childbirth? Is that a good one? Is that a pro or a con? That's a con. Okay. That's a con for sexual, right? Okay. Just gotta be careful. Gotta make sure.
I know, you're just.
Childbirth, right? Partner, what is that? So where does it go? Don't need a partner. Right? So by contrast, that would be a con for sexual, right? You need to talk somebody into it. Sounds a little manipulative, right? It's like this is anything else? What?
If you have something wrong with you, then.
So that would actually, so you're talking about a genetic diversity issue, right? So no genetic diversity. I mean, it is, but that's mostly because of the organism itself. That's a little tricky to deal with because there's multiple variables in there that you have to parse out that we really can't. So in terms of genetic diversity thing, that would be what? A pro for sexual, right? So in this case, you would have lots of genetic diversity, maybe, or more. Okay, anything else?
For pros,
Yeah, cons, either one.
well, I heard in humans and dolphins, it's
So what would that be? A pro for, okay, pleasure fact. So that would be what, a con? So no pleasure. Mhm. Any others? Think in terms of energetics, efficiency. So that would be what, a pro?
more energy
Where?
than sexual.
That is a con? So it takes too much energy for sexual and less energy, right? For asexual. Yeah. And one would argue an entire lifetime after that. Yeah. So you're on the hook for this one, right? Okay, so when you're looking at this list, is it me or is there a little bit of a lean toward asexual?
Asexual is when you
Think-
do it with yourself, right?
No, it's like you're just making a carbon copy of yourself. Yeah.
I heard some lizards like that.
We have quite a few organisms actually do asexual. So, right. But if you think about it, think about the themes we've been talking about biology, right? Or we just got done talking about the benefits of having low energy state, right? The cell is always looking for a good deal, right? Maximum benefit, minimum energy cost, right? So if you're looking at this from the energy frugality lens, which one are you picking?
Oh, one more pro for asexual. They said it's like it's exact, like you don't have to split your genes because of that. So it's like it's just you. You know what I mean?
So you don't need a partner.
Well, that, but because like if you have a kid, it's your genes and your partners. But if you're asexual, it's just like it's almost like a
Yeah, a carbon copy. So you don't need a partner. And it's a replica of you, so they're exact. What, by the way, no genetic diversity. Can you see that as a con? I mean, as a pro, right? So if it's good genes, right, copy is good.
With it, with that being said, less chance for similar in the sense where.
Assuming you have good genes, right? But Kira started off with saying, well, if you've got this train wreck in your genome, then it's going to compound the train wreck, right? But the flip side of that, I'm saying, right? The flip side of the genetic diversity thing is it could be good, right? I mean, if you've got good genes, then asexual is the way to go, right? So here's the question. Just looking at this list, if you had no idea about biological diversity on the planet, wouldn't you kind of assume that you would expect to see at least roughly about half and half, right? Because there seems to be good arguments for both. Maybe slightly leaning toward asexual because it's a little bit more efficient, less costly. But you know what you actually see? It's overwhelmingly sexual. Interesting.
This size, energy, size,
Well, it does. Like, I mean, you have to be multicellular to be sexual. So you can't be a unicellular. So at some point, you have to have some sizable body to be sexual. Oh, the diseases, yeah, STDs. Right, that's true, right? So you don't have that with asexual. Don't have to worry about that one. So it seems to me this is a little on the heavy side down here.
Crystal inbreeding.
Yes, right. There's some more. Well, you guys are really negative here. Got a bunch of celibates here. Just kidding. It's like, no. Right. Wait, say that again? Sure. Like a mutation or something like that? Yeah, mutation can happen either way, either direction. So here's the thing though, right? We start to see some really heavy negative for sexual reproduction. And yet what we see out there is a world that's really tilted heavily in favor of sexual reproduction. So think about it. Think about the way the cell thinks, right? Nothing comes for free. So if you're willing to lay down Ben Franklin's on the table as a cell, what are you demanding in return? You're demanding value. You're demanding value on your money, right? It's like, okay, I'll lay down a lot of money, but man, you had better give me some titanic value for that. So when you see a lopsidedness like this, especially with all these negative drags on sexual reproduction, whatever the benefits are, the pros, these things had better be massive, right, to counterbalance that kind of cost. And we know it's a cost, don't we? Because think about it. I mean, let's just talk about this one here. How easy is it to talk somebody into this? Not so much, is it? It sounds straightforward, but it's not so easy, is it? I mean, we got an entire industry based on this, don't we? Match.com, eHarmony, the entire dating scene. Yeah, Vegas in general. Right? I mean, a good chunk of our existence is talking somebody into it, right? And that's just us. I mean, other animals are the same way. I mean, you've got the little peacock strutting around with his pretty little feathers. Why is he doing that? He's looking for the girl, right? I mean, the two dudes at the beach pumping iron, one's trying to sort of outflex the other. What are they doing? Yeah, they're trying to basically say, look, my muscles are bigger than his, so you know you want me. That's exactly the same thing, yeah, that animals do, right? Notice though, this is an entire spectrum in animal biology and zoology that we refer to as mate selection. A lot of times we hear about natural selection. That's easy, right? That's just you get killed because you're fluorescent pink, right? And the predator sees you first. But a lot of times what has a lot to do with especially genetics is mate selection. Is how does an organism go about choosing their mate? Not every single one is like, oh, I want the one with the biggest, prettiest feathers, right? So each species has a slightly different criteria. By the way, notice that this mate selection is a big deal too. How do you know that? Because that dude who's really brightly colored, like the birds are notorious because we love birds because they're colorful this time of year, especially they get their spring plumage because it's mating season. And so all the dudes are all pimped out in their tuxes, you know, whatever that looks like. And they're all looking good. But guess what? Birds are what's called sexually dimorphic, right? So the two sexes have a different plumage coloration. The dudes typically tend to be pretty, and the girls, well, not so much, right? So they're camouflaged. But when you see something like that, sexual dimorphism, a lot of times students will anthropomorphize. They're like, oh, sexist birds. Like birds aren't sexist. It's not possible for them to be sexist. They're birds, for crying out loud. They're not sexist. This is just their biology, right? So biologically, when you see sexual dimorphism like that, their biology has been arranged so that the hen is the primary caretaker of the offspring. And of course, if you're a hen laying on eggs and those eggs hatch and you got these little naked, defenseless little baby chicks under there, the last thing you want to be is fluorescent pink that has a giant sign on your head saying, eat me, I've got defenseless chicks under me. This is one of the reasons why the females tend to be camouflaged and the males colorful. But then what are the males doing? They've got the sign that says, eat me. So natural selection is against heavy coloration, but they do it anyway, don't they? Which tells you how strong mate selection is in counter to that. Right. So that just gives you an idea how big of a deal this is and how much energy this takes. So here you're talking about an organism where a huge chunk, a swath of their biology is consumed with just this one piece right here. I have to talk somebody into this by being as pretty as possible. Betsy. Chickens, no, they're sexual. And they're also sexually dimorphic. Because you can pick out a good rooster by coloration and his little punching bags on his head and his chin. And hens are usually, I mean, they're not like non-colored, but their colors are slightly different. So, and you can see a lot of times the roosters will have like, the little ruffles, and they got extra little frills and things like that in their plumage for females as well. Well, yeah, you've already done it. I mean, there are some organisms who do that. Then there's some organisms who are tight, and they will constantly mate as long as you let them. So it depends on the organism, right? Okay, so what's the benefit then, right? So how come in the face of all these negatives, we've talked these negatives up a lot, then why do we all do it, right? What's the payback? It's very simple. Gets back to this one. The name of the game is variation. And in order to explain to you the importance of variation, genetic variation, what I want to do is in a bit of an analogy. So I'm going to write an exam and I'm going to give it to Jared. He didn't know that, but I'm going to give it to him. But I will also give him the key. So he's got an environment, the exam, and he's got the answer to that environment, the key, right? So then I basically tell Jared, okay, pass it on. So he's going to pass it on to Rainey, right? And then she's going to take the exam as well. So does she change anything? No, because Jared didn't only get a 90. He got 100, right? Because he's got the answers to the problem. So when he passes that on, do you change anything? You just fill out the key, right? Because then pass it on to Melissa. Do you change anything? No. Now pass the exam on to Christopher. Now I'm going to change it. I'm going to mess with you. Yeah. So now what I'm going to do is I'm going to take the exam away from, I'm going to give a different one. But it's not a bio exam. It's like musicology or music theory. It's like political science of the rise and fall of the Whig Party. Okay, so you know what the Whig Party is. But, yeah, what is the Whig Party? No, just kidding. No. Right, so here basically you have something you're not prepared for, right? So assuming that you don't know, like let's say you were an immigrant and you don't know hardly anything, just the basics about American history, but certainly nobody has any clue what the Whig Party is. The average American doesn't. Most of you guys look at me like, the Whig Party? You mean we had a Whig Party here? Yes, we did. But anyway, long story, not worth, it's a pretty dry story actually, but it's an important one if you want to understand the Civil War. But he has the key and he has a new test. But guess what? He has nothing else. So how do you think we're going to do on that test? Right? Because the key is answering a different test, isn't it? So he really has two choices. He can guess and hope he gets enough guesses in his favor to pass. That would be like if you're asexual, that would be like I can mutate and hope I get enough mutations to get me through this environmental change. What's the likelihood of that happening? Since you get a beneficial mutation, maybe what, once every 500,000 years, that's pretty generous. This is not a strategy for you, is it? So what's most likely going to happen to you? You're going to die. In your case, you'll fail the exam, right? Because you're not, why? Because you don't have enough variation in your head to be able to give you a shot at this new exam. What is that variation? It's knowledge. So now let's say I change it. So that's the asexual pattern. But now let's say I change it. Now I have Christopher read this book by Michael Holt on the rise and fall of the American Whig Party. Big book, great book. It's you got to be a poli-sci major really to appreciate it, right? But basically he reads through this whole book, learns everything about the rise, the beginnings of the Whig Party, the end of the Whig Party, and right up to before the Civil War. And then I give him the exam. So now is it a little bit different? Now he's got a shot at the exam, doesn't he? Because now what happens is because when he comes to a question he doesn't know, it's like, well, I don't really know the answer to that one. What he does have is a deep reservoir of knowledge that he can dig into. That reservoir of knowledge is like genetic variation. The deeper your reservoir of genetic variation, the more likely you can go in there and pull something out that's going to help you answer that question correctly. That's sexual reproduction. So literally, if you think about it, this is a story between life and death.
Martin Van Buren in the Whig Party?
Martin Van Buren was not the Whig Party. He was the enemy of the Whig Party because he was Jackson's vice president, and he was the Democratic Party, which was different than the Democratic Party of these days. But the Whig Party was largely anti-Jacksonian. The Whigs hated Jackson. And Van Buren was basically just a Jacksonian puppet, essentially, is what he was. They did, yes. They were the ones that ended slavery, not the Democrats. Yeah. Okay, so when we take a look at sexual reproduction, the name of the game is variation, right? So essentially what's going to happen is we're essentially going to be going through genetic information that is coming from two different individuals, mom and dad, right? And this basically results in obviously genetics, which in the short term is just basically genetics. That's like chapter 12, we're studying genetics. But in the long term is evolution, right? So when you think about it, evolution is a genetic idea. Why? Because evolution is the change in species over time. How do species change? They change by changing their traits, and their traits are governed by genetics. So evolution at its core is and always has been a genetic idea. This is one of the reasons why, in my perspective, in my opinion, evolution honestly doesn't work when you try to view it through the lens of the rock zoo, better known as paleontology. That's why I always say, and I'm very clear about it, I've never been impressed by people digging out a bunch of bones from rocks, especially when you're trying to argue evolutionary relationships, because it's like you trying to say, well, let me find a couple of people. Crystal and Unit. It's like me trying to say both of you guys are related, but you're not, are you? You know, for a fact. But I'm saying both of you guys are related. Why? Because what I'm looking at is features. You both have dark, long hair. You both have dark eyes, similar skin tone that's relatively in the ballpark. So therefore, morphologically, you guys must be related. Are they related? No. That's the kind of evolution that we had for well over a century, which is the reason why I'm not impressed by the rock zoo and everything that comes with it. Because now genetically, when we take a look at you guys, we can clearly see, no, your genetics is coming from a different family lineage than your genetics, which is a completely different family lineage, right? So I didn't understand that relationship, and I couldn't claim to understand that relationship until I understood that within the lens of genetics. This is the reason why I'm not a big fan of 19th century and 20th century evolution, because they basically view it completely incorrectly. Those are hard words for somebody who came from UC Davis. I could hear all my UC Davis professors screeching because they're just in love with the morphology, when in reality it's genetics, not morphology. So when we take a look at sexual life cycle, this is essentially what determines all the biology of organisms, right? So basically, we've already kind of been down this road a little bit. We sort of have fertilization, where the sperm of one individual male who is carrying a haploid set of chromosomes, right? So basically 23 chromosomes, will fuse with the egg that also has 23 chromosomes, and you're going to get your diploid number, which is having two sets of chromosomes, right? So basically, you have your diploid number, which is our full number, and of course your haploid number, which is N. So that basically is half that number. So you only have one. This is going to be largely located in your gametes, whereas the diploid number is going to be located in your body cells, right? So that's basically what we have. And then, of course, we get this information from both our parents. So essentially, the sperm of dad, which has 23 chromosomes in it, will basically fuse with the egg of mom, which has 23 chromosomes in it. The male pronucleus will fuse with the female pronucleus, reconstituting the zygotic nucleus of 46 chromosomes at this particular point. And then off we go to build our body. By the way, if you didn't catch it, you weren't paying attention, you probably would have missed it. But what you just got was the really quick 30-second elevator rundown version of the birds and the bees. Okay. Just saying, act surprised when you get there. Yes. I love this chapter, but anyway, so now let's take a look at life cycles. Actually, one of the things you do in zoology is before you study a new taxonomy, like a new phylum, you start with their life cycle, their sexual life cycle, because everything they do in biology is centered around the sexual life cycle. Now, in some organisms, like in animals, most of our living existence is in the diploid state. Your body is a diploid, right? So if you walk in the bathroom and you take a look in the mirror, what you're seeing is mostly diploid. The only cells that we have that are haploid are the germline cells, the eggs and the sperm. That's it. So we are mostly dominated by the diploid state. That's not true for some organisms. Like some organisms will alternate between a haploid body and a diploid body. This is what's called alternation of generations. And so here's what we look like, right? So we're going to start off with the zygote. So the zygote, which is diploid, will go through mitosis to create your diploid body. And of course, that's going to go through mitosis to mature to your full adult reproductive body. In this case, in the ovaries, the female will send a subset of cells to send them through meiosis to create the egg, which is a haploid. The male will take some cells in the testes, and then we'll send those through meiosis to create the sperm. Upon fertilization, the two of these will fuse together to reconstitute the zygote of the next generation. But notice, the overwhelming majority of your lifespan is actually spent in the diploid state. The actual cells themselves that are haploid last for a very short period of time. The egg, for instance, lasts for maybe what? A month, right? So basically the female will ovulate the haploid egg, where it'll sit and wait patiently for fertilization. And if it doesn't happen, then the uterine lining, which at that point has become very vascularized and thickened, the internal uterine lining will realize, wait a minute, okay, so I don't have an embryo coming my way anytime soon, right? So that's the reason why it vascularizes and thickens up, is it's an expectation of implantation of a newly fertilized embryo. When that doesn't happen, and then basically detaches, sloughs off along with the egg. So you lose the egg as well. And then you start it all over again. So you queue up the next round of follicular development to create the next egg for ovulation. So at best, the egg, at least in the ovulation cycle, the follicular development cycle, is around for maybe a month-ish or so, right? Sperm, on the other hand, are even less long-lived. The average male will make a fresh batch of sperm roughly every 24 hours or so and make a bunch of it. But sperm outside the body doesn't last for long at all, because it doesn't have anything in order to actually maintain its life, right? So cells need a nucleus where they can make proteins, they need organelles to do all their business, but all sperm is basically a little genetic bomb. That's all it is. It's a little genetic grenade. It's just DNA, that's all that's in here, with a little protein capsule and a little protein flagellum with a little bit of mitochondria, just enough to keep it motile. But those mitochondria are like little wind-up toys, right? So you're kind of like the little wind-up parts of it, and they're just basically sort of unwinding, and that's kind of what gets the sperm to sort of swim. And then each mitochondria will just basically peter out and stop, and then eventually they'll all stop, and then the sperm will die. Okay. That doesn't take very long at all. Okay. So a lot of our life is spent in this diploid state. That's not true for some organisms. Let's take a look at moss, right? So moss is in the plant kingdom. It's kind of like what we refer to as protoplants. It's what we evolutionary envision is sort of like what came before plants. So before gymnosperms, cone-bearing trees like pine trees and things like that, and flowering plants like angiosperms, and roots and all that sort of stuff that we notice that we associate with plants. Before all that, we had mosses, right? So a moss basically will have two different bodies. So one body that it'll have is a diploid body called the sporophyte. And then from this, you'll create a little sporangium, which has a little capsule or a pod in it. And then it'll have certain cells in there, which will go through meiosis, creating haploid cells. And then eventually this little pod will open up. And then out comes a little haploid spore. Now, this little haploid spore will land in some dirt somewhere, and it'll go through mitosis. And many rounds of mitosis, and it'll create a haploid body. So this is your haploid body called a gametophyte. Then what's going to happen is a certain subset of cells of the gametophyte will go through not meiosis because it's already haploid, but it'll go through mitosis, and it'll create the haploid gamete. And it'll fuse that to another haploid gamete from another individual, and then they will reconstitute the diploid zygote, which will then go through rounds of mitosis to create the sporophyte. A little more convoluted, but you have, and when you take a look at a moss, two visible bodies, either a gametophyte or a sporophyte. Now, that kind of sounds weird, doesn't it? If you wanted to see what this looked like, and it kind of is a little confusing a lot of times for students until we take a look at it with respect to us, right? So if this happened in us, this is what it would look like. we would basically have a zygote that would go through mitosis to create our body, right? This would be like our sporophyte. So that's the same. Then we would have a mature body, which then produce haploid cells, just like the spores, right? So that's the same thing here. So we have the same thing so far. But now instead of fusing to go through fertilization, what happens if our gametes actually went through mitosis? So the sperm would go through mitosis, the egg would go through mitosis, the sperm would create like a little half clone of the dad, and the egg would create a little half clone of the mom. So you'd have this new generation of half clones running around. And then they would go and live their life and do whatever they do. But then what would happen when it came time to finish the reproduction is that the half clones would come together, form gametes through mitosis, and then they would fuse their gametes together to create the zygote, which would then grow up to become the new sporophyte, the new diploid body. Kind of weird, isn't it? So basically what happened is you yourself wouldn't reproduce. You would create half clones running around, and then your half clones would run around the world, and then they would be the ones who would finish the reproduction. So your grandkids would be the ultimate outcome of your reproduction, right? Now think about what a mess this would be for us. But we'd have this entire group of humanity that were half clones. You could almost imagine all the causes and the laws, right? You know, half clones are people too. Right. I mean, you can hear all these laws, you know, equal rights for half clones and right protection, equal protection under the law for half clones. Just because you only have half a genome doesn't mean you're half a person, right? I mean, you can hear the bumper stickers, right? Because why am I saying that? Because that's exactly what we would do, right? If we had a world of half clones, we would treat them like non-human. That's probably what we do. That's just what we do. Unfortunately, we do it to ourselves, right? Right, we do it to ourselves when we're fully human and fully equal. I mean, so we would probably do monstrous things to our half clones. Is that a question? So they would only carry half of your genome. So they would only have like half of your traits. So they probably would look different. So what would be the reason for this? Well, why would you do this in the first place? Well, the reason why you do this, now we deal with it differently, right? The reason why you do this is very easy, very specific, right? So let's imagine that, say, for instance, when we went to go have kids, a lot of times some of us will delay that decision, right? We'd be like, you know what? I want to have kids, but I don't want to have it right now. I'm young still. I want to do some college. I want to get a career. I want to make some money. And then once I kind of get myself together, then I want to have a family, right? So what are you doing? I want to improve my circumstances. I want to find a more suitable and a more beneficial environment for my kids once I get to have them, right? So what we do is we just delay that time frame. But for a moss, it doesn't do that. What it basically does is says, listen, the circumstances I'm in right now aren't so great. So I would like my offspring to have a better environment. So what I'm going to do is I'm going to create these little spores. I'm going to cast my spores out throughout the world. Some of those spores will be in very, very favorable places. And then they can finish out the reproduction and take benefit and advantage of all those extra better environments that I can't get to. Right. So you spread your spores around so that you can take advantage of better environments, more adaptive environments. It's a survival strategy. So let's take a look at meiosis. So with meiosis, basically you have one division that has two sections to it, a meiosis 1 and a meiosis 2. They all have basically the same process as mitosis. They have a prophase, metaphase, anaphase, telophase. I'm going to talk about synapsis later. But when you take a look at meiosis, the two different steps of meiosis, first meiosis actually have very specific things that they do. The first one is what's called the reductional division. And the goal of the first one is to basically separate homologs. So that's what they do. They're basically separating the homologous pair from each other. The second meiotic division, meiosis 2, this is what's called the equational division. Because what you're doing here is you're going to separate your sister chromatids from each other. Very similar to what you did in mitosis, okay? Now notice what happens is in between, there's no DNA replication. You're not talking about two different cell division events. You're talking about one cell division event that's broken up into two stages, Okay, that's the reason why you don't have replication in the middle. So let's take a look then, first of all, what's happening, right? So you break up your meioses in like prophase 1, metaphase 1, anaphase 1, telophase 1, of course, prophase 2, metaphase 2, anaphase 2, telophase 2. Notice what's missing. Prometaphase. Remember, this is the reason why I said that sometimes you see a 4-phase versus a 5-phase system. This is because in meiosis, you're not interested in microtubule dynamics, which is what's happening in prometaphase. You're interested in chromosomal dynamics, right? So as a result, you see prophase, metaphase, anaphase, telophase. You don't really focus in on the events of prometaphase. Does that mean prometaphase isn't happening? No, it's all still happening, right? It's just that that's not the focus of meiosis. Meiosis, you're focusing on chromosomes. So let's take a look and see what is occurring in these stages. Let's start off with prophase 1. So in prophase 1, we have basically some of the same things that happens in prophase of mitosis, right? So some of those things are basically chromosome condensation. So that's completing. That's also happening in mitosis, yes? We also have spindle migration. Is that happening in mitosis? Yep. That's happening in mitosis too. We also have nuclear envelope breakdown. Is that happening in mitosis? Yeah, so what's different then, right? Why is meiosis such a thing? Well, let's take a look at the things that are unique to meiosis. First of all, you have homologous pairing. And then after you pair the homologs, you have synapsis, and then after synapsis, you have recombination. So what does that look like? So essentially what you get is you get your 2 chromosomes together, mom 1 and dad 1. You're going to go through replication, and then you're going to pair your 2 together. So this is mom 1 and dad 1. Now in homologous pairing, the 2 homologs find each other. Mom 1 finds dad 1, and you zipper them together with a structure called the synaptonemal complex. This stage right here, when they're homologously, they're synapsed together, this is what's oftentimes either referred to as the tetrad, because you've got 4 chromatids, 1, 2, 3, 4. Or you hear it referred to as the bivalent or the bivalent, because you've got 2 homologs zippered together. Terminology wise, you can hear those. Then what happens is you form these little crossover points, these exchange points between these, which are driven by what's called chiasma. These are crossovers or recombination, areas of exchange between these 2. But what I want to do is I'm going to actually talk a little bit more about recombination, because there are 3 major engines that we see actually in meiosis, what I call engines of variation. Recombination is the first engine of variation. Think about your engines of variation. There's 8 billion people on this planet. There will never be another you. There never has been another you. There is not another you. There will never be another you. Do you believe that?
Never.
Never. Just from what we understand with variation, do you believe that there's enough variation in genetics to make that to be true? So what we're going to do is we're going to drive that point home, right? So in order for that to be true, that basically means that you are lightning in a bottle, that you will never be captured the way that you are in your body at this time twice, right? And so what we're going to do is we're going to take a look at recombination. We're going to take a look at a chromosome. We're going to take a look at dad 1. We're going to take a look at mom 1. Now, dad 1, here's your chromosome. So there's your one sister. There's your centromere. There's your other sister. And then a mom 1, here's one sister. Here's your centromere. Here's the other sister. Now, what I'm going to do is I'm going to start off the recombination event. It always starts off by a double strand break, right about here. Now, there's 2 ways that you can heal this double strand break. you can either join the two ends together like this. If you do that, you'd get nothing, right? Or because they're homologous, you can actually join the end of one homolog to the end of the other because they're the same. So you can take this purple one and drop it down here and join it to the pink one like that. This is a chiasma, chiasmata for plural. A physical exchange or a crossover between these two. I'm going to go ahead and draw two different exchange points here. I'm going to draw this a little bit better. There. And then what I'm going to do is I'm going to go ahead and fast forward this, and we're going to take a look and see what our meiotic products are. So the first thing we're going to do is we're going to go through meiosis one. We're going to separate our homologs. We're going to separate them like this, okay? And then we're going to take it through meiosis two. We're going to separate the sisters individually, okay? So let's take a look and see what that looks like. So when I start off with this particular one here, what I'm going to do is I'm going to basically pick this up. I'm going to go all the way through, pick up the purple centromere and all the way to the end. So this particular chromosome looks like this. Now let's take a look at this one. This one starts off purple, but then as soon as I hit that chiasma, I go across the bridge, and then I pick up the pink, pick up the centromere, then I hit the next crossover, then I go across the bridge, then I pick up the purple, and I finish it out. So this one here looks like this. Now for number three. For number three, you pick up the pink, you hit the crossover, you go across the bridge, then you hit the purple, then you pick up the centromere of the purple, you hit the crossover, you go across the bridge, and then you pick up the pink. So this one here looks like this. Now let's take a look at four. Four basically is all pink. Pick up the centromere, and it looks like this. So now you have four haploid, genetically non-identical daughters, right? Isn't that what the outcome of meiosis was? Four genetically non-identical daughters. Now think about it. Numbers 1 and 4 look like the parents, right? 4 looks a little like mom 1. 1 looks a little like dad 1. So these are what's referred to as the parentals. 2 and 3 look like a combination of the two, right? These are what's called the recombinants. They've been shuffled. So basically then, this is your gamete. So if you're a male, these become 4 sperm. If you're a female, you usually pick one of these and that becomes the egg, okay? So do you think now, 8 billion of us and moving up, and potentially trillions more of us, depending on how long we live on this planet, do you think this is enough variation to make that claim? But there's never been a you on this planet. There's not another you on this planet, and there will never be another you on this planet. Is that enough variation for you? Some of you are happy. You guys are very agreeable. Right? My Monday, Wednesday sections were not so much. I had to really prove it to them. Chaos theory. Oh, but let's make it worse, shall we? This is just one cell going through one meiotic event. If you're a male, this is just 4 of your potentially trillions of sperm. By the way, let's take a look at this. Let's kind of make this a little bit more random. These guys right here, the crossover events, their position where they land, where those double strand breaks land, is random. So what is the likelihood of getting a crossover event exactly right here and right here twice? Not likely, right? Not only that, but guess what? You can have one crossover, 2, 3. So the number of crossovers you have is also random. So you have 2 layers of randomness, the number of crossovers and the position of crossovers that add to the shuffling. So just looking at the recombinants themselves, it's virtually impossible to get the exact same breakpoints between 2 recombinants twice. It's like lightning hitting the bottle twice, right? It's nearly impossible the first time. It's mathematically impossible the second, right? So, but we're not done yet. Because usually when people push back on this and say, there's not enough variation here to explain that I'm the only one in 8 million, right? Because what they see is they see this, right? Because they say like, well, because this is not variable. I mean, this looks exactly like the mom chromosome, and this looks exactly like the dad chromosome. So really, if you think about it, half of what your outcomes are really only shuffled. The other half are like, they look like carbon copies to me. That's not variable. That's not variation. Oh, but it is. You know why? Because this chromosome here is not all pink. The chromosome that your mom had when she went through meiosis and she shuffled them up to give to you or not, the chromosome she started with, the pink one, itself was shuffled by grandma, which was then shuffled by great grandma and great grandpa and shuffled by great grandpa and great grandma. And this is the biggest problem with genetics, because a lot of times in genetics, what we do is we tend to think of genetics in the near term. We tend to think of our generation, the generation before us, and generation after us. So we only usually think about 3 generations mostly in genetics. But in reality, the chromosomes that you are holding are like a chimeric quilt of every single individual that has ever added genetics into your lineage, going all the way back to before the dawn of time. So every generation will take what they've been given, shuffle it, and give it to the next generation, which will then take it and shuffle it and give it to the next generation, which will take it and shuffle it and give it to the next generation. So on my chromosomes, I hold little bits and pieces of genes that came from somebody who may have lived in Germany. Or here's a good one, Africa, right? This is the reason why 23andMe is so fun, because people always get a percentage out there, right? And they're like, totally white. I mean, as can be, like a snowbank, right? And then they get 23andMe and they say like, well, wait a minute, there's a percentage of my ancestry that's African, right? And so then they take a look at it, it's like, oh, wow, well, what that means is somewhere in your family lineage, somebody from that continent married into your family, the genetics are there. And then over time, through shuffling and shuffling and shuffling and shuffling and shuffling, then what you see is, like these little shards, like little genetic shrapnel of all these different groups and people groups that have married into your families. So each of these is basically a wild kaleidoscope of stuff, right? So there's a lot of variation. As a matter of fact, that's kind of what genetics is like. It's like taking a puzzle, like taking half of it, because you only put forward half of your genome, right? And you glue it all together. And then what you do is you recut the puzzle and you pass that forward. And then that generation takes half of it, glues it all together, and then recuts it. So basically what you have is a jigsaw puzzle that's been jigsawed, that's been jigsawed, that's been jigsawed, that's been jigsawed so many times that that the pieces don't even make sense anymore, right? It's just like you just have like these little shards, these little bits and pieces. Okay. I love to think of genetics as sort of like those pictures that we see that are made of pictures. Have you seen those? right? Like if you look closely, they're like little bitty tiny pictures that kind of makes up the person. That's exactly what chromosomes are like. It's like when you stand back, you see your family, your immediate family, you can see the resemblance. But then when you look really closely, you can see there's all these little itty bitty pictures from all these different places that you didn't even know like existed, right? And so that's kind of what genetics is like. So here's the example. This is just a comparison of meiosis 1 and mitosis. So here you can see meiosis 1 is going to be lined up in double file. That's important, right? Versus single file. Why? Because when you're lined up in double file, you get your exchange here, your recombination. Then when you basically move, you're going to be moving your homologs in either direction. Notice you have your two parentals here and your two recombinants here on the inside. Now you choose to separate those guys out. Mitosis, nothing, right? You basically take your carbon copy sisters and you basically pull them in two opposite directions. So much simpler process. So crossing over then basically recombination is essentially a very, very powerful engine for genetic variation. So you can get a lot of information. And actually you can see, some of us actually have this in our families, right? Where you can kind of see that sort of progressive shuffling in our families. Sometimes some people call it dilution, genetic dilution, but that's basically just kind of shuffling it out. And a good example is like taking a family that is largely dominated by light features, right? Like a family with a lot of light hair color, light eye color, light pigment color. And then like if somebody who marries into the family who has dark features, dark hair, dark eyes, dark skin. And then you can see that from the next generations as those dark features start to get shuffled forward, right? So in the immediate generation, you can see kind of like a mixture of the two genes going on, right? The dark versus the light going on. And we see this a lot in interracial families. But then as those generations go forward, you can see it kind of gets a little more diluted out, depending, of course, on who they marry, right? But let's say, for instance, that it was other light individuals that kind of came in. But what you can see is kind of this progressive generation by generation, it kind of like gets a little lighter and lighter and lighter. But guess what? The genetics are still in there, right? Because all the dark genes for pigment and hair color and skin color are all still in there, but they've been sort of meiotically recombined. They've been shuffled around from generation to generation. So they're still in there, right? And every now and then what will happen is you'll see them crop up, because then you'll see them come into play. And it's like, oh, here's this darker the rest of them, right? It's like, okay, where'd this come from? It's like, well, it's always been there, because back a few generations ago, you had this infusion of traits that came into the family. And that kind of actually happens in my family, because I mean, most of the people in my family are just like me, snowbank, right? I mean, they're just like super white. But a few generations ago, my great grandma had all dark features. She marrying into a German family, which was a light-haired, light-eyed, light-skinned family, but she had all dark features. So she had dark skin, dark eyes, dark hair. I think she was like a Native American by descent, of something of that nature. But the dark features dominated our family for a long time. Like, as a matter of fact, that's the reason why my dad has black hair and dark eyes, and same thing with his dad. But then in this generation, because we kept getting reshuffled, right? Then what happened is it moved forward, and then we have the first generation where we're kind of lightening up a little bit. But my middle daughter is actually, you can kind of see that skin tone coming back out, because the skin tone, that darker skin tone, although it was gone for a good long time, just all kind of came together in my middle daughter. So she's like perceptibly more olive toned in skin color than her siblings. And we all look at her like, wait, where'd you come from? It's like, well, I kind of do have this kind of Native American ancestor that married into the family a few generations ago. And that's part of where it's coming from on my side, you know, even though technically I don't have a lot of that to show, it's still kind of all in there. Your chromosomes still have these little snippets in there from these families. And every now and then, given the right compounding factors, they can come in and reinforce. And we see that every now and then, don't we? We see a kid that kind of is a surprise. It's like, whoa, where'd you come from? It's like, well, it's always been there. It's just, there's been these little chunks that finally you put enough of them together and genetically you got it. And so that's kind of what we have with crossing over. And so here you can see you're crossing over. So you got basically your two homologs find each other. They're still held together by cohesins. Remember the cohesins, these guys are holding the sisters together. And then of course they cross over, and then your synaptonemal complex is going to be in here in the middle holding your homologs together. And then when you separate these guys out, you can see you've got your four products, your four genetically non-identical products. So that's kind of what it looks like in prophase one, right? So we have our standard stuff. So we have our spindle pole migration, right, which is happening. We have our condensation, of course, and our nuclear envelope breakdown. But now we have the homologous pairing and synapsis, and of course the crossover part of it, right? So that's all new. And that's all the innovations of meiosis. Most of the innovations of meiosis are happening in prophase one. So that's what these are. So what's going on in metaphase then? Well, in metaphase, actually, what we have is basically the same basic idea, right? So from metaphase, so the spindle poles are attaching, only now they're attaching to the homologs themselves, right? So they're attaching to the kinetochores of the homologs. And these homologs are lined up side by side, right? So they're in double file, and that's important because that's going to make sure that you get the proper segregation into the two different piles, right? The separation of your homologs. And so that's kind of what it looks like. So here you can see your spindle has basically organized itself. You still have the same thoroughfare, right? Those anaphase B microtubules that are attached to each other. In this case, now you've got your spindle attached to each of the homologs, which at this point are both recombined. You can see that they're still tangled up to each other, and they also have the synaptonemal complex in between them. And the important thing to understand about that synapsis is that whatever you do to synapse these two homologs together, you have to resolve it when it comes to anaphase. So now not only do you have cohesin to resolve, this will resolve in meiosis too, right? But now you have synaptonemal complex. Which gets resolved in anaphase one. Synaptonemal, SYNP, synaptonemal. It's an O. Synaptonemal. SYNPTONEMAL. Yeah, no, none of these science jargon things. Your spell check is just going to hyperventilate and spontaneous combust on you. Right, so basically you have to resolve that when you get to anaphase 2. And so that's basically what's holding all these guys together. But notice you have this double file alignment. But let's take a look at how you align. This brings us to our second genetic variation engine. We have our first, right, recombination. This is our second. It's called random assortment. Think about it. You've got 23 unique chromosomes, right? 22 autosomes that are all lined up with each other. 1, 1, 2, 2, 3, 3, and so forth, all the way up to 22. And then you got X and Y lining up with each other, or X and X lining up with each other, right? So you got 23 rows of chromosomes. But think about it. Is there anything that determines where you put mom versus dad? So what's the chances of mom falling on this side? Well, what's the actual number? Yeah, it's 50-50, right? It's 1/2. It's a coin toss. That means dad over here is also going to be coin toss, right? That means it's equally likely that mom could have ended up on the other side. Literally, you're tossing quarters. But think about this for a second. You're tossing quarters 23 times. Right? That's an easier probability one to do that, right? How many combinations can you come up if you toss a quarter 23 times? So you can do that, right?
23 times.
Well, you have two different outcomes, right? So your probability is 1/2 to the 23, right? So that's a lot, right? So basically, you have 23 separate events, each of them with a 1/2 probability associated with it, right? So that's basically what you're talking about. So essentially what you have here then is you have multiple different possibilities. So you could put mom, mom, mom on this side, right? Or you could do mom, mom, dad, or you could do dad, 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 or you could do dad, dad, mom, or you could do mom, dad, mom, or mom, dad, dad, or dad, mom, dad, or dad, mom, mom, and so forth, right? So we're only doing 3. There's 23. That makes sense? So how many combinations can you generate just from this random assortment? A lot, right? Does that give you variation? Sure it does, right? Because if you're this one right here, then that means this cell over here is going to get predominantly mom chromosomes. This one over here is going to predominantly get dad chromosomes, right? So in some cases, this could help you. It's a little more tricky than this actually, but what did it come out to? 8.3 million. So that's a lot, not infinity, right? But it's a lot, especially when you add this as a second layer already on top of recombination. So this could also explain, well, there's a little bit more nuance to this. The reason why sometimes we see like a family lean in some people, right? Have you ever heard the term, you take after your father, right? Usually it's not a good thing, right? Right? Or, you're just like that. And we see that, don't we? Genetically, we see that sometimes a sibling will have a greater resemblance or a lean toward either the maternal side or the paternal side of the family. What could possibly explain that? Well, this could simply be that your maternal resemblance to the family could simply be that you had a bit more maternal chromosomes or a random assortment lean on your side. Right? It's just that simple. It'd be obviously different for this one, which is going to have a lot of dad in it with a little injection of mom, or this one's going to be mostly mom with a little injection of dad in there, right? So you can kind of have a mixture of these different outcomes. So each one of these is different. And it's a coin toss. And this is before you actually add in the recombination. So this is your second major genetic variation engine. No, I don't think so. I mean, theoretically you can. The problem with that one is if you can trace it back to your original ancestor, I mean, you really are getting to a collision of worldview at that point, because whether or not that's possible would only be true is if you had kind of that biblical kind of Adam and Eve perspective, in which case it should be possible to do. But if it's more of an evolutionary type of a scenario, then it's not going to be really that possible, because there isn't an Adam and an Eve, unless there is an evolutionary bottleneck, which we do know that there have been human bottlenecks in the past, in which case the survivors of the bottleneck would be sort of like that proto-Adam and Eve scenario. So, yeah. But oftentimes in evolution biology, we do use the term Adam and Eve for the survivors through bottlenecks like that that became the genetic forebears of kind of a new world. So it's helpful to think of these genetic engines as random number generators. And so what we'll do is we'll pick up with anaphase next time.
I heard if the world was the basketball game, humans would be in like the last fraction of the set. I heard LeBron.